Now, I would imagine this is going to take you probably about 2 to 3 minutes. You're just going to put everything down that you could possibly think of. Like for me, I would have probably started off by drawing. To me, I would be a drawer because I like to model. So I would draw a cell, and I would draw a nucleus, and then I draw the DNA inside it. Okay. And then maybe I'd make a list of a couple of things that I know that's true about it, which is we're going to review in just a second here. Okay. So we know that DNA, if I was to get this out of our way here, we know that DNA is made up of nucleotides. If you recall that one thing that Dr. Deamer had, where you had the 4 quadrants of proteins, carbohydrates, and lipids, those were the 3 macromolecules in foods. And then the 4th component was your DNA, and your RNA was nucleic acids. Well, if you remember, the outer ring stood for the polymers, and the next ring N stood for the monomers. Well, nucleotides, remember, are the monomers of nucleic acids, DNA and RNA. We know that a nucleotide has 3 parts, a phosphate, and we can see, I don't know why it looks kind of silly here, but it's going to be some sort of phosphate molecule. It really wouldn't look like a circle. You would see that it would have a phosphorus, and you've got a couple of different other atoms attached to it. Right now, it's not important to you, but it actually would be a small molecule of that. Oops, let me go back here. And then you would see that here you're given a sugar. You're noticing that if we're dealing with DNA, this would be a deoxyribose sugar, right? Remember, the sugars are different. And we know that each one of these corners is representing, if you recall, where a carbon atom would be present. And then we can see that it's being attached to a nitrogen base. Now, what are the 4 bases of DNA? Do you know what those are? Take a moment and write them down. What are the 4? And there's 4 letters. Did you get them? Did you know that you're dealing with adenine, thymine, cytosine, and guanine, right? ATCG. Okay, so adenine, thymine, guanine, and cytosine. And again, this would be an individual nucleotide. That's a monomer of a nucleic acid. In this case, our nucleic acid is DNA. And we can see the 3 parts of it, okay? And you'll notice that it doesn't make a difference, cytosine, guanine, adenine, or thymine. They all 4 have the same sugar, which is a deoxyribose sugar, and they all 4 have a phosphorus molecule. Again, this looks like it's just going to be a small circle. That's not really what it looks like. It's going to be an atom connected to other atoms, but for right now, we're not worried. Now, for your DNA to make that beautiful double helix, okay, for it to be able to be produced, and I get myself out of the way here, we can see that to make this beautiful double helix, that's the term that they gave for it, okay, you can see that there's this outer portion that we often refer to as the outsides of the ladder, okay, those are the support parts, if you will, and that's the part that's going to be made up of the phosphate and the sugar. And so you can't see it here very well, but over here you could see that this would be that one side of the ladder, and this would be the other side of the ladder, okay? And so you can see you have a phosphate, a sugar, a phosphate, a sugar, a phosphate, a sugar, a phosphate, a sugar, okay? And over here you can't see where it would be, but I do know that the sugar is what's attached to the nitrogen base. So I know there'd have to be a sugar molecule there. Again, you can't even see it from this because they've got all the nucleotides attached. But there would have to be a sugar right here, and then there would have to be a phosphate here. If this is showing this direction, sugar, phosphate, sugar, phosphate, sugar, phosphate. And then over here would be phosphate, sugar, phosphate, sugar. So phosphate, sugar, nitrogen base, phosphate, sugar, nitrogen base, phosphate, sugar. This just isn't breaking it up into those individual parts. But you can see, oh, by the way, we oftentimes refer to this as the sugar phosphate backbone. And we'll often refer to this part where you have your hydrogen bonds that are connecting all these together. We often refer to these as the rungs of the ladder or the steps, okay? And that's where the nitrogen bases are going to be connected. This would be a visual of DNA. And we know it's DNA. Why? Because it has thymine in it. Remember? DNA has thymine, but RNA does not. We'll see in a second here. Okay. So the order of these bases determines the DNA's instructions or the genetic code. Now, if we look at this, this is kind of cool. We're just giving you a little example of how we can use this molecular information to help us figure things out. What we have here are two small sections of the I gene for both a mouse and a fly, okay? So you can see the nucleotide sequences there. And you know it's the DNA because you can see thymine in there, so you know it's all DNA. But they're the exact same gene. They're the exact same gene. In other words, when you look under that double helix and you locate where it is, because we know where the chromosomes are. We know where everything pretty much is located. And so when they look at that and then they compare the nucleotide sequences, it's pretty amazing. But you'll notice that the sequences are almost 77% similar. That's pretty cool. So for the same type of, or part of that, of a fly and a mouse, the I gene, okay, we're pretty much seeing the exact same thing. It's 77%. That's huge. That's really, really, really big, okay? So what do we care? Why do we care about all this stuff? Well, first of all living things are made up of DNA, okay? So it doesn't make a difference if it's bacteria or fungi or plants or animals, kind of like we were talking about before. The function of DNA is to store that genetic information that codes for traits. And examples of those traits down below here, I don't know if you can see mine, but mine are attached. So you can see here that I have what's referred to as an attached earlobe. Well, it's really kind of interesting because when you look at all of my sisters, they have a detached earlobe. No wonder my sister used to tease me all the time. Anyways, but the key thing is that what's going on here? Well, when you look at my parents, you might see what they are, and they both could be detached. Then you're like, well, then how did you become attached? You ever heard of the term carrier? Maybe my parents are carriers. And so if both of them are a carrier of an attached, then there's a possibility that I would become an attached. Same thing happened with my blood type, by the way. I'm the only one in the family that has O type. Everybody else is A. If we look at here, there are some individuals that have to deal with color challenges, okay? They can't see this really deep red, okay? They don't have those, oh shoot, I'm having a brain freeze. Is it the cones or is it the rods? Brain freeze, I'll have to go back. If you guys are in my classroom, you'd be able to tell me. But the bottom line is there's one of those components that's been damaged. And so because of that, this is what they're gonna see and set. And I gotta tell you, that'd be really challenging as a driver because you'd have to make sure that you were paying attention very carefully since we use red and green, okay, for our light structure. And then left-handedness and right-handedness. My husband was left-handed, I'm right-handed, but he had broken his left arm in a motorcycle accident. So he taught himself how to write really beautifully with his right hand. So he could do either one. He became ambidextrous. And then of course, dimples. Look how cute that little dimple is. And some of you do. Some of you in my classroom have really great dimples. And so again, these are what DNA would be storing the information for those individual traits. And you would display them. Now, when we say display them, well, there's two things. If we're looking at the genetic information, which we are in a minute, when we're looking at all those nucleotides and sequencing them, and we're looking to see how they connect, that's the genotype. When you're seeing what you actually look like, that's your phenotype. Best way for me to remember those is genotype has genes, phenotype, I think of photograph. And so, phenotype is what you're actually going to visually see. Genotype is what the genes are saying is going to transpire, okay? All right, so why do you look like your family? Well, it makes sense, okay? Genes are inherited. They're received from someone that came before you. And so genetics is that study of inheritance. So if one parent had, in this case, blue eyes and the other one had green eyes, okay, then the colors, the possibility of getting a brown would be 0, okay? Being a blue would be 50 and being a green would be 50, okay, it'd be 50-50. DNA is found, and what's so key about this, in every cell in every living thing, every living thing. And so when you take that cell and you create that nucleus and you look where that DNA is, okay, when you were doing all of that, you found that when the DNA is being requested to make something up, it's going to have to duplicate itself. Like for instance, if you're going to split the cell, if your body cell, which is a skin cell, is going to make another skin cell, well, the very first thing that's going to have to happen, which is pretty much what happens the majority of the cell's life, is that it goes on and it takes in all the nutrients necessary to make more and more and more. And then what it does is, at first, it's going to replicate. It has to duplicate the DNA first so that it can split. Because if every cell has got to be identical for a body cell, then, like a skin cell, then that would mean that you would want the DNA to duplicate and then split. And then from there, a lot of other stuff goes on. But when we talk about chromosomes, that's when your DNA condenses, and it does so, and it coils up. And you're going to see there's a really cool video that we're going to add to this. Either have it in the resources on Canvas, or it'll be connected into here. But it shows you how those, how this incredible length of DNA can fit inside every single one of your cells. It's truly mind-boggling to me. It's just phenomenal. And so then you're going to see there's parts of your DNA, of your chromosomes, your DNA, whereas if you were to unstretch it, okay, and so you uncondense it, okay, if you uncondense it, this is what it's going to look like. If you condense it, it starts to make the chromosomes. And then you're going to see it's got some special parts to it. We'll get into those parts later. Centromere is maybe that center portion, and then the rest of these we'll get into later on. So every single cell in your body has an entire copy of your DNA. Pretty amazing. DNA is found in the nucleus of the cell. That's the DNA. Oops. Okay, so that's that. Okay, so then this video, you're going to want to take a moment and you're going to want to stop and watch this. So, oops, Daisy, let's go back. Go back. Okay, so if I come up here, I'm going to stop recording for just a moment so you go and you watch the video.